Imagine you are trying to repair a commercial jet engine, but the plane is currently in a steep nosedive.
Right, not an ideal situation.
No, definitely not. And normally, our baseline understanding of evolution says you just have to wait for millions of years of, random, incredibly slow genetic mutations to maybe, just maybe, fix that engine.
Yeah, it's a cosmic lottery.
Exactly. You just have to hope the environment doesn't destroy you before your species randomly mutates the exact right DNA sequence.
And that slow, meticulous chipping away at the genetic code, I mean, that is the textbook definition of adaptation. Permanent, random changes locked into the DNA.
But recent discoveries are completely upending that textbook.
Oh, entirely. What we're looking at today is the biological equivalent of the cell possessing a hidden panic switch.
A panic switch.
Yeah. A way to temporarily put down the fine chisel, grab a sledgehammer to survive that nosedive, and then go right back to normal later, as if nothing ever happened.
And unpacking that secret switch is the mission for our deep dive today. We are exploring this absolutely mind-bending 2026 paper from the journal Cell.
It's a massive paper.
It really is. It's titled Prion-based Protein Self-Assembly Tunes Mutagenesis to Enable Rapid Adaptation. So we are going to look at how cells use a specific kind of protein origami to temporarily rewrite their own mutation rates.
And the craziest part is the tool they use to do this. Prions.
Right. Usually you hear the word prion and you immediately think of terrifying, incurable conditions like mad cow disease.
Exactly. We think of them as these broken, toxic proteins that misfold, clump together, and just destroy brain tissue.
So, okay, let's unpack this. How could a toxic misfolded protein actually be a secret weapon for survival?
It sounds totally counterintuitive, I know. But this research reveals that specific prions actually function as highly evolved, heritable mutagenesis switches.
Okay.
They allow organisms to rapidly adapt to lethal challenges. So everything from toxic chemicals in a lab to the actual antifungal drugs we use in hospitals.
Right.
And the kicker is, they do this without permanently altering the baseline DNA sequence. It's a temporary, reversible hijacking of the mutation rate.
Which is wild. So to really grasp how a clump of proteins saves a cell, we have to look at the initial experiment these researchers set up. They used yeast, right?
Yeah, they took 28 different strains of yeast, and each strain contained a different known prion.
And in the paper, if you read it, you'll see these prions written in a very specific way. It's a bracket, italics, all caps, and a plus sign.
Right, so bracket prion plus.
Yeah. And that notation is crucial, right? It signifies non-Mendelian cytoplasmic inheritance.
Which is a heavy term.
It is a heavy term, but it basically just means this trait isn't being passed down through the classic DNA in the nucleus. It's being passed down through the actual physical shape of proteins floating out in the cytoplasm.
Exactly. So the researchers wanted to see how these prion-carrying yeast strains would react to extreme chemical stress. They exposed them to a proline analog called AZC.
Let's define that real quick. Proline is an amino acid, right? A building block of proteins.
Right. And AZC looks just like proline. So when the cell tries to build a new protein, it gets tricked. It accidentally grabs the toxic AZC, inserts it into the chain, and the resulting protein misfolds massively.
They basically feed the cell garbage parts.
Basically, yeah. And they also hit the yeast with tunicamycin.
Tunicamycin, okay.
It's a nasty chemical that disrupts end-linked glycan biosynthesis. To put it simply, it jams up the cell's protein assembly line in the endoplasmic reticulum.
So it causes a catastrophic backup of unfinished, unfolded proteins.
Exactly.
So if you jam the assembly line and trick the cell into building garbage proteins, you would expect the yeast to absolutely panic. Alarms blaring, mass deployment of molecular chaperones, which are the cell's cleanup crew, right? Rushing in to fix the toxic mess.
That is the logical assumption. But when the researchers performed mRNA sequencing on 32 different strains to see exactly which genes were firing, the chaperone response was shockingly quiet.
Wait, really?
Yeah. Out of all the emergency cleanup proteins available to the cell, only 3 showed any statistically significant induction.
Only 3.
Yeah, we saw a chaperone called HSP26 go up in one strain and SSA2 go up in 2 others. That's it. The massive protein folding panic we expected just wasn't there.
But the cell wasn't just sitting there dying, right? Something else was quietly activating.
Right. In 20 out of those 32 strains, the sequencing revealed a massive alteration in the expression of genome fidelity factors.
Okay, so instead of trying to clean up the misfolded proteins, the cells were turning on and off the genes that controlled DNA repair, DNA replication, and the checkpoints that monitor cell division.
Exactly. They were rewiring the control room.
I like to think of a prion like a piece of sticky molecular Lego. Usually you worry that a sticky Lego will just randomly clump up with other pieces, jam the cell's engine, and break the whole machine.
Right.
But what this sequencing shows is that these specific sticky Legos are intentionally changing their shape to go interface directly with the engine's control panel.
That's a great way to put it. They are rewiring the DNA replication system, not breaking the cell.
So how did they measure the impact of that rewiring?
They used a brilliantly elegant tool called the Luria-Delbruck fluctuation assay.
Okay.
It's a classic method for measuring spontaneous mutation rates. They took these prion strains and exposed them to a toxic arginine analog called canavanine.
And this is where the puzzle gets really cool, because canavanine is lethal to yeast.
Very lethal.
But the yeast can only absorb it if they have a working importer door, right? Which is built by a specific gene called CN1.
Right, so if the yeast randomly mutates and accidentally breaks its own CN1 gene, the door disappears.
And the toxic canavanine can't get in, and the yeast survives.
Exactly. So by counting how many yeast colonies actually survive the poison, you can calculate exactly how often the yeast is making mistakes in its DNA. You are measuring the mutation rate by measuring survival.
That's genius. And what did the assay show?
Well, the data showed that 13 out of 46 prions tested caused stable, heritable changes in that spontaneous mutation rate.
Wow.
And we aren't talking about microscopic shifts either. The mutation rate swung wildly, anywhere from 2-fold up to 15-fold in both directions.
So some prions cranked the mutation rate up, making the cell highly error-prone, while others dialed it way down, making the genome incredibly stable.
Exactly.
Okay, let's take a quick breather and synthesize what we've discovered so far. Here's a bullet summary for you to lock this in. First, prions aren't just toxic accidents. They are actual functional, heritable switches within the cell.
Right.
Second, instead of triggering a generic stress panic when things get bad, they specifically rewire the expression of DNA repair genes.
Crucial point.
And third, we know from that Luria-Delbruck assay that they act like physical dials, drastically turning the mutation rate up or down.
Which brings up a really interesting question for the audience.
Yeah. Think about your own life for a second. Have you ever realized that a flaw or a mistake in your routine was actually a hidden feature keeping you adaptable? That's what the cell is doing. It's weaponizing its own mistakes.
It is a profound shift in how we view biology.
Right.
But, knowing the mutation rate changes is only half the battle.
Right, the mechanism.
Yeah, the essential question is the physical mechanism. How does a clump of protein floating in the cytoplasm actually reach in and turn the dial on DNA replication?
So let's zoom in on one of those dials. The researchers looked closely at a specific anti-mutator prion, meaning it dials the mutation rate down, making the cell more accurate.
Right.
It is called bracket POL32+, and it's formed by a templating protein naturally called POL32.
And POL32 is a fascinating piece of machinery. It is a non-catalytic component.
Meaning it doesn't do the chemical heavy lifting itself, right?
Exactly. But it acts as a structural bridge for two completely different DNA polymerases. And polymerases are the machines that copy your DNA.
Okay, lay them out for us.
First, POL32 is part of POL delta. That's the high-fidelity replicative polymerase.
So that's the careful one.
Yeah. POL delta is made of POL3 and POL31, and it is the careful, meticulous copyist you want handling your genome.
Okay.
But POL32 is also a structural component of POL zeta, and POL zeta is made of Rev3, Rev7, and POL31. This one is a translesion synthesis polymerase.
And that's the messy one.
It is so sloppy, so error-prone, that geneticists literally call it the mutism.
Wait, hold on. I'm stuck on something here. If the POL32 protein misfolds and clumps up into this prion state, isn't it basically a broken tool?
It seems like it, yeah.
So if a component of the DNA copier breaks, shouldn't the copier make more mistakes? How does a broken machine result in a genome that is suddenly making fewer errors?
What's fascinating here is that the protein hasn't lost its function. It's actually a change in localization.
Localization.
It changed its zip code. The researchers proved this using fluorescent tagging. They attached a green fluorescent protein, or GFP, to POL32.
Okay.
When you look at normal, naive yeast cells under a microscope, that green glow is diffuse. It's spread evenly all throughout the nucleus, which is where the DNA lives. The copy machines are in the office doing their work.
Right. But what happens when the cell switches to the bracket POL32 plus prion state?
In 75% of those prion cells, the glowing green POL32 completely vanished from the nucleus. Yeah. Instead, it formed concentrated glowing dots, which we call puncta, in the cytoplasm, entirely outside the nuclear membrane. It physically relocated.
Okay, imagine the error-prone mutism, Paulzetta, as a crew of incredibly messy, blindfolded painters.
I love this analogy.
In a normal cell, they wander into the house, which is the nucleus, and occasionally splash paint all over the DNA walls.
Right.
But when the cell triggers the prion state, that clumped protein acts like a massive biological bouncer. It grabs these messy painters by the collar and physically locks them out in the yard.
Exactly.
Because they are sequestered in the cytoplasm, they literally cannot reach the DNA to paint it. So without the messy painters in the house, the genome suffers fewer mistakes. The prion becomes an anti-mutator simply by changing the location of the risk.
Spatial regulation is incredibly effective. And the evolutionary depth here is staggering. This physical sequestration trick is not just an anomaly in baker's yeast. No. The human homologue of POL32, the exact equivalent protein in our own bodies, is called POLD3. And previous research has already shown that human POLD3 can form these exact same heritable protein assemblies.
Wow. So we share this spatial regulation strategy with yeast, meaning it is an ancient, deeply conserved biological mechanism.
Exactly.
Okay, before we move on to the next prion, let's do our recap to lock down that mechanism. Bullet 1. We've got the bracket PLO32 plus prion acting as a massive anti-mutator.
Lowering mutation rates.
Bullet 2. It achieves this not by destroying the mutism, but by acting as a bouncer, physically sequestering the error-prone copiers outside the nucleus.
Right.
And bullet 3. This mechanism is conserved all the way up to human proteins. It's a brilliant strategy.
It really is.
So here's a question for you to mull over. Where else in society or in your own life do we solve complex problems simply by moving a risky element into a different room entirely?
That's a great question.
Yeah.
But yeah, there's a flip side to that coin. If the bouncer locks down the genome to prevent mistakes, what happens when a cell actually needs new traits?
Right. What if the environment suddenly becomes so toxic, so deadly, that playing it safe means certain extinction? If you are in that jet nosedive, you need new traits instantly.
Enter the diversifier. The researchers investigated another prion, originally named bracket MPH1+.
Okay, MPH1.
It is formed by the MPH1 protein, which stands for mutator phenotype 1. MPH1 is a helicase.
Basically a motor protein that unzips the DNA double helix, right?
Exactly. And it is deeply involved in interstrand cross-link repair, or ICL. ICLs are essentially toxic superglue that locks the two strands of DNA together so they can't be copied. MPH1 usually unzips and fixes that.
Given the name mutator phenotype, you might expect this prion to just blindly smash the DNA, causing random base pair mutations to desperately find a way out of the crisis.
You'd think so, yeah.
But wait, if you just start taking a sledgehammer randomly to your DNA, doesn't the cell just die? How is this prion actually generating useful diversity without just shredding the genome into garbage?
Well, the cell is much smarter than that. The researchers discovered it doesn't just create random, raw, double-strand breaks. Instead, this prion does its most important work during meiosis.
Which is the cell division process for sexual reproduction.
Right. During meiosis, chromosomes line up and swap pieces of DNA. The MPF1 prion dramatically hypercharges this recombination process.
Oh, wow.
Yeah, it takes the existing genome, fragments it into smaller haplotype blocks, and intensely shuffles the genetic material. It creates offspring with extreme, highly varied combinations of traits. It was so effective at mixing things up that the authors actually renamed the prion to bracket MEX+.
That is so cool. The analogy that comes to mind is a high-stakes card game.
Let's hear it.
If you are losing badly, random mutation is like trying to win by drawing on blank cards with a marker. You are probably just going to ruin the cards.
Right, totally untested.
But what bracket MEX+ does is entirely different. It is like taking a loaded, proving deck of cards and performing a flawless, radical shuffle to get the best possible hand.
In genetics, this is called the reassortment of standing variation. You aren't inventing new tools from scratch. You are brilliantly recombining the tools you already know work.
Just putting them in a radical new order.
Exactly. And to confirm this was a universal strategy, they looked at the human homologue of MPH1. It's a critical repair protein called FANCM.
Okay.
They use a brilliant dual plasmid system to test it in yeast. They inserted one plasmid, which is a little circle of DNA, that produced human FANCM tagged with mRuby.
So a red fluorescent protein.
Right. Crucially, they put this red FANCM under an inducible promoter. That means they could turn it on or off with a specific chemical trigger.
Okay, got it.
Then they added a second plasmid, producing FANCM tagged with GFP, a green protein. but they put this one under a constitutive promoter, meaning it is constantly churning out green protein no matter what.
So they have two separate supply lines of the exact same human protein, just painted two different colors. What do they do with them?
They chemically triggered the red FANCM, creating a massive overabundance of it in the cell. This spike forced the red FANCM to clump together and form the prion assemblies.
Okay.
Then, they removed the chemical trigger and diluted the yeast cultures by a factor of 1,000. They basically washed away the red protein, letting it naturally fade from the population over several cell divisions.
But the green protein was still there, constantly being produced.
Exactly. And even long after the red protein was entirely gone, the green FANCM protein continued to form large, enduring fluorescent foci under the microscope.
Wait, so the temporary pulse of the red protein successfully templated the shape of the green protein?
Yes. The physical shape was passed down like a molecular ghost, proving that the human FANCM protein retains this exact same prion-like self-templating memory.
A temporary pulse creating a permanent, heritable memory. That is mind-blowing. Okay, let's do our subtopic recap. Bullet 1. Bracket MEX plus acts as a genetic diversifier.
Bullet 2. It works by increasing meiotic recombination without increasing raw, lethal DNA damage.
Perfectly shuffling the deck.
Exactly. Bullet 3. It shuffles existing traits to help offspring survive extreme stress. So here's your question for this section. If you were facing a massive crisis, would you rather invent a completely new, untested tool or brilliantly recombine the proven tools you already have?
I think nature clearly prefers the latter. Nature is ruthless and pragmatic. But if we connect this to the bigger picture, we realize this is not just a quirky trick that baker's yeast does in a petri dish.
Oh, definitely not.
No, this mechanism is an active, highly lethal battleground in hospitals right now.
The clinical stakes here are massive. Fungal infections kill millions of people globally every year, and they are rapidly developing resistance to our frontline drugs.
Right. And the researchers didn't just study safe lab yeast. They secured clinical isolates of yeast from actual patients, including the World Health Organization priority pathogen Candida albicans.
What did they find? These clinical pathogens naturally harbor these exact prion switches. To prove they were relying on them for survival, the researchers set out to cure the yeast of its prions.
How do you cure a prion?
They used two methods. First, a chemical called guanidine hydrochloride. Second, they used a dominant negative version of a protein called HSP104.
HSP104.
Yeah, it is essentially the master factory manager chaperone of the cell.
I want to dig into that factory manager role. Why is a chaperone, a protein whose job is usually to unfold clumps, essential for maintaining a prion? Shouldn't HSP104 be trying to destroy the prion?
It seems contradictory, right? But HSP104 is actually the engine of inheritance for these prions.
How so?
As the prion fiber grows long and sticky in the cytoplasm, it can't easily be passed to a daughter cell during division. It's too big. So HSP104 acts like molecular scissors. It comes in and chops the long prion fibers into smaller seeds.
Oh, wow.
Those tiny seeds are then distributed to the daughter cells when the yeast divides, where they start growing and templating all over again. Without HSP104 chopping them up, the prions don't get inherited, and the cell is cured.
So they fire the factory manager, they inhibit HSP104, stopping the seeds from spreading, effectively curing the clinical yeast strains of their prions. What happened to their mutation rates when they did that?
The rates swung wildly, losing their adaptive edge. In one specific clinical isolate from a patient called F16256, the mutation rate plummeted 38-fold the moment it was cured of its prions.
38-fold. That is a massive loss of genetic flexibility.
Exactly. And this is specifically how Candida albicans survives our hospital drugs. Candida relies entirely on this HSP104 manager to evolve rapid resistance to fluconazole.
Which is one of our best antifungal weapons.
Right. When the researchers tested Candida with its HSP104 inhibited, the pathogen suddenly hit a brick wall. It completely failed to adapt to the drug. It couldn't evolve.
Because it lost the physical mechanism required for massive resistance.
Right. Normally, when Candida is hit with fluconazole, it survives by generating copy number variations, or CNVs.
Let's define CNVs for the audience.
Essentially, it acts like a photocopy machine with a jammed button, duplicating huge chunks of its own genome. We are talking pieces of DNA that are 20 kilobases or larger.
Massive chunks.
Yeah, it mass produces the specific genes it needs to pump the drug out of its system. But without HSP104 to maintain the prion state, the pathogen completely lost the ability to trigger these massive genomic duplications.
So by targeting HSP104 in the hospital, we aren't actually trying to kill the fungus directly with a stronger poison. We are actively turning off its ability to evolve.
We are drugging evolvability.
Drugging evolvability. That is a brilliant phrase. Okay, let's summarize this clinical application with our final recap. Bullet 1. Clinical fungal pathogens actively use prions to rapidly develop drug resistance.
Yep.
Bullet 2. HSP104 is a chaperone protein essential for chopping up and maintaining those prion seeds.
And bullet 3. Inhibiting HSP104 prevents the massive genomic changes, those CNVs, needed to survive antifungals.
So here's your question to think about. What if the future of medicine isn't just creating stronger poisons to kill bugs, but simply freezing their ability to adapt to the poisons we already have?
It is a totally new front in medicine. This research highlights a massive shift in how we understand biology. We are looking at epigenetic memory achieved through protein self-assembly.
Right.
Cells can temporarily tune their genome plasticity. They can lock down their fidelity when times are good, ensuring perfect copies, and then hit the genetic shuffle button when times are deadly. And they accomplish all of this without ever permanently breaking their underlying DNA sequence.
And that leads us to a final, really provocative thought to leave you with today. We mentioned earlier that the human proteins PLD3 and FANCM retain this exact prion-like ability.
We did, yeah.
But there is a darker side to those proteins. We know from analyzing massive human cancer databases that mutations in these specific proteins often map directly to what are called their intrinsically disordered regions.
Which is chilling, because intrinsically disordered regions are the flexible, unstructured, floppy areas of a protein. And biologically, those exact floppy regions are the necessary ingredients required for a protein to misfold and form a prion.
It makes you wonder, could human tumors be using these exact same ancient shape-shifting prion switches to outsmart our chemotherapy?
Are the cancers that seemingly evolve overnight to defeat our modern medicine just exploiting a 300-million-year-old survival program?
It's a terrifying but absolutely fascinating idea. We started today talking about evolution as a slow, methodical sculptor with a fine chisel. But it turns out the sculptor has a sledgehammer hidden under the workbench, and it knows exactly when to swing it. Thank you for joining us on this deep dive. Go look at the world of protein folding in a whole new light.